Human beings have their own cells outnumbered by the bacteria in their body by approximately a 1:10 ratio. The relationship between humans and the bacteria that inhabit their bodies is more complex and important than previously thought. The science of having a healthy microbiome is now getting more attention, as many publications now show that our normal microbiome not only helps us digest foods and produce vitamins like B2, B12, and K, but also helps provide a barrier to pathogens or even attack them directly, along with modulating our innate and adaptive immunity. One of the great threats to humankind is the rise of antibiotic resistance. In the United States, it is estimated that over 2 million illnesses due to antibiotic resistance, with about 23,000 deaths annually. Yet, many patients still demand antibiotics, and many medical professionals are willing to oblige. Antibiotics not only kill susceptible pathogens, but can kill the commensal bacteria that serve as a natural protective barrier. Overuse of antibiotics can also confer resistance on commensal bacteria. These resistance genes can potentially be shared with a future pathogen. Antibiotics have a huge impact on the microbiome. Aggressive use of broad-spectrum antibiotics given to premature infants, generally ampicillin and gentamicin, has been associated with an increased risk of late-onset sepsis, primarily caused by group B streptococcus, necrotizing enterocolitis, and an overall mortality and selective advantage of drug-resistant microbes. Antibiotics have been shown to alter the transcriptome and proteome of host tissues. The positive association between antibiotic use and subsequent celiac disease suggests that intestinal dysbiosis may play a role in the pathogenesis of celiac disease. Antibiotics, with their impact on the human microbiome, have shown to increase body mass index in patients treated with vancomycin and gentamicin used for infective endocarditis or antibiotics for treatment of Helicobacter pylori infection. The microbiome is the ecological community of microorganisms and their corresponding genetic information that lives in a particular environment. Each person has their own microbiome that they depend on. The relationship between people and microbes occurs in several different relationships. 1. Commensal. In this relationship between two organisms, one benefits and the other gets either no benefit or harm. As an example, Staphylococcus epidermidis lives on the skin of individuals, so we provide it a place to live, but we do not get direct benefit from it living there. 2. Mutualistic. In this relationship, both organisms benefit. As an example, certain Escherichia coli in the gut can produce vitamin K while humans give it a place to live. 3. Parasitic. In this relationship, one organism benefits at the expense of another. As an example, malaria is caused by Plasmodium being transferred from a mosquito bite. It infects liver cells and then red blood cells, causing damage to host individuals. And 4. Opportunistic. Some bacteria can have commensal or mutualistic relationships with the host at most times, but can become pathogenic when the appropriate conditions arise. An example of this is Streptococcus pneumoniae that frequently colonizes children, but can then cause diseases such as otitis media and pneumonia. The human body has a host of different environments that can harbor different microorganisms. Different regions of the body can vary in many ways, including oxygen level, pH, temperature, and nutrient sources. As such, different areas of the body harbor different microbes. Many hospitals test the nares for potential methicillin-resistant Staphylococcus aureus or MRSA, since that particular bacterial species likes warmer, moist areas. Yet we see similar environments in the armpit and groin, so patients may be negative in the nares and still enter a hospital carrying MRSA. Microbiome is sometimes used to mean all the microbes in a community, or sometimes used to denote the full collection of genes of all the microbes in a community. It is estimated that there are about 20,000 human genes, yet that pales in comparison to the 9 million genes that the bacteria in our body can carry and how they can potentially turn on or off our own genes. The understanding of the microbiome has evolved over time from paying attention solely to pathogens that need to be eradicated, to understanding that most human or bacterial relationships as being one of necessity. The mature adult microbiome has been assessed across many populations. The largest project in this area to date is the Human Microbiome Project, which assessed 15 to 18 body sites in 242 participants in 2012 and continues to sample new individuals. An important finding from this project was that microbial populations differ substantially among healthy individuals, and so far no single microbial composition has been defined as healthy, aside from a preponderance of bacteroidetes and firmicutes. The human microbiome encompasses several trillion microbes residing in the body, particularly the gut, and the genes that are encoded by them. The majority of these microbes reside in the colon, where they are present in a concentration of up to 10 exponent 12 CFU per milliliter and play a crucial role in the functioning of the digestive tract and in harvesting energy from the diet. The microbiome maintains the integrity of the intestinal epithelial barrier, thereby offering protection from pathogenic bacterial colonization. It is essential for metabolizing indigestible polysaccharides and in the absorption of short-chain, fatty acids produced by bacterial fermentation. The microbiome also helps in the regulation of intestinal transit, thereby affecting the amount of energy absorbed from the diet. The initial microbiome of a child is established during birth where the mother's bacteria can be transferred. Before birth, babies are somewhat sterile. When they are delivered through the vaginal canal, they are exposed to microbes from the canal along with skin and fecal microbes. Babies born by C-section may have more skin cell bacteria. The disruption of this transmission during birth has been linked to celiac disease, asthma, and type 1 diabetes in some studies. To mimic the colonization that occurs in vaginal birth, some doctors are placing gauze in the mother's vagina and then rubbing it in the mouth, face, and body of babies born by C-section. The microbiome can be further established by breastfeeding. Not only does breast milk contain the microbes themselves, but prebiotics or food that is non-digestible by a person, but can be by certain bacteria, that can help promote the growth of particular microbial species. Diet has an impact on the microbiome as demonstrated by De Filippo et al., who saw an increase in prevalence of bacteroidetes and depletion in firmicutes in children from Africa who were consuming high-fiber diet as compared to children on high-sugar or high-fat diets. Likewise, some bacterial species unique for fiber degradation like Prevotella and Xylanobacter were absent in the children from Europe consuming high-sugar or fat diet. Additionally, in a recent study of antibiotic-naive children in Niger, it was shown that a single dose of oral azithromycin was enough to affect the diversity of gut flora when tested 5 days later. According to current estimates, 600 million people around the world are obese. Diet, inaction, and genetics have all been commonly cited factors in weight gain. However, additional experimentation is pointing to the microbiome of individuals playing a significant role. In mice experiments, one study showed that obese and lean mice had differing floras. When the gut flora from an obese mouse was put into the germ-free mouse, it became obese. In this experiment, humanized mice or mice carrying functioning human genes or tissue, when given the intestinal flora from an obese twin became obese and the flora from a thin twin stayed thin. The diet between the two did not change. When these mice were then put in the same cage, the obese mouse lost weight, presumably due to fecal transfer of bacteria from droppings. In further studies, when a large amount of bacterial strains are given to an obese community of mice, they shift to become lean. However, if bacteria is transferred and then the mice are given a Western diet which are high in fats and sugar and low in fruits, vegetables, and fiber, the obese mice stayed obese. In an additional study, mice that were given antibiotics in low doses had 15% more body fat and less microbial diversity than the control mice. These findings are suggesting that a high diversity of gut flora is linked to better health. However, this diversity needs to be sustained with the proper diet. Affecting mammalian behavior is Toxoplasma gondii. It is an obligate intracellular parasite that is capable of infecting most warm-blooded animals. In the normal life cycle of T. gondii, oocysts are shed into cat feces. These can sporulate and infect intermediate hosts, such as rats and mice. Cats are then infected through the cysts of the intermediate hosts and the cycle begins again. If people are infected, there are often no symptoms as the immune system keeps this in check. The concern here is for pregnant women as the infection can be passed to the baby. The child can be born with eye or brain damage or potentially develop blindness or mental disability later in life. For this very reason, pregnant women should not clean cat litter boxes or eat undercooked meat. When T. gondii infects mice, within 3 weeks, it loses its fear of cat odor. As such, this parasite is able to alter the host's behavior in such a way that it critically lessens its possibility for survival. Researchers were able to develop a strain of T. gondii that could mount an immune strain that caused T. gondii to be eliminated from the mouse. After 4 months, it was not detectable in the brain of mice. Yet, these mice still did not fear cat odor, which suggests that this parasite has the ability to make permanent changes in the brain. Toxoplasma gondii has been linked to high-risk and less self-control behavioral changes in people. In humans, many people are now calling the gut the second brain. The genome of the gut microbial community has been hypothesized to be 100 times larger than the human it inhabits. There is almost 200 square meters of mucosal tissue that is entirely colonized by a microbial community in each individual. It is now well established that there is bidirectional communication between the gut and brain via neural, endocrine, immune, and humoral pathways. Researchers are now postulating that particular microbes are actually able to regulate brain function. Studies are now being done looking at the gut flora normally found in obese people and seeing if there is a link to disorders of the central nervous system, such as depression. In another study, attempting to see if there is a link between microbiome and mood, an experiment was set up to give mice low-dose penicillin in late pregnancy and early postnatal life. Some mice received Lactobacillus rhamnosus JB1 to help recolonize the microbiome. The results of this experiment showed long-term changes in gut flora with differences in the integrity of the blood-brain barrier. Brain cytokines were increased in the frontal cortex and these mice exhibited anxiety-like and socially impaired behavior. The control group receiving Lactobacillus rhamnosus had some of the alterations prevented. There is also evidence of indirect changes in behavior based on microbial flora. A child diagnosed with pediatric autoimmune neuropsychiatric disorders associated with streptococcal infections, exhibited obsessive-compulsive disorders and/or tic disorders that are associated with group A streptococcus or gas infections. Their hypothesis is that gas tries to evade the immune system by engaging in molecular mimicry, so that it looks like host cells. However, if a susceptible child develops antibodies, they can cross-react to self-antigens. These symptoms can resolve when the child is treated with antibiotics, but may return if the child is recolonized in the future.